Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar. I'm Melissa Manzotti, CEO and founder of Bio-Insights, and today we'll be discussing how to plan for success, emerging GMP-grade raw materials and technologies for cell therapy. As more cell and gene therapies become approved for clinical use, there's definitely been a rapid increase in the need for innovative cell manufacturing technologies that can really help improve product efficiency and maintain cell product integrity, meeting the scale-up demands. To overcome this commercialization challenge, it is really important to identify process development workflow stages where optimization is necessary. Test and qualify new technologies that can improve the different stages of the workflow, and importantly, establish supplier relationships to really secure raw materials scale up. As part of our starting materials spotlight this month, I am absolutely delighted to be hosting the webinar today with a panel of leading experts who can share their real-world experience and guidance on this critical topic. First up will be a presentation by Sean Kevlahan, Senior Director of Cell and Gene Therapy at Bio-Techne. Sean is a product-driven entrepreneur, a specialist in cell biology, and domain leader in cell and gene therapy bioprocessing. Prior to his role at Bio-Techne, he was a CEO and co-founder of Quad Technologies, which was recently acquired by Bio-Techne. Following his presentation today, he will be joined by our expert panel for a Q&A session, during which we would, of course, love to hear from our audience members. So please do feel free to post a question in the Ask a Question box, and we will very much try to get to it during the course of the webinar. And so with that, I will hand over to Sean.
Well, thank you for giving me the opportunity to speak. My name is Sean Kevlahan. I am the Senior Director of Cell and Gene Therapy at Bio-Techne. And today I'm going to be talking about optimizing workflows for ex vivo cell therapy manufacturing. Before I get started, I'd like to give a little color on who Bio-Techne is and what we're all about. So Bio-Techne offers a cornucopia of reagents within research as well as diagnostic and GMP reagents. You might have heard that we have started a new cell and gene therapy initiative from pioneering cell and gene therapy solutions from discovery to the clinic. And herein, we're actually coalescing a lot of our high-quality products, including products from R&D Systems, Novus Biologics, Tocris, Protein Simple, and ACD, into one clear message around cell and gene therapy manufacturing, again, going from discovery reagents and tools to clinical manufacturing. Our workflow solutions are focused on quality and consistency. We take a unit operation approach to developing products through patient monitoring, cell separation, cell culture, and in-process quality control and in-process analytics. Our products that are offered within this workflow follow strict regulatory guidelines, including USP 1043, 92, and European standards for raw materials to help support cell and gene therapy manufacturing. Consistency is a really important metric for us, and we've invested heavily within ISO 9001 and ISO 13485 facilities to help translate our research products into GMP products to ensure consistency. And we leverage a mature QMS system to help with consistent manufacturing and quality. Now, as you might know, a workflow process optimization is necessary to achieve scalability. Process development is a crucial step with manufacturing cell and gene therapy products, and you really need to evaluate your workflow process to achieve scale. And so there are some key parameters that you might want to look at, such as yield, efficiency, phenotype, and reproducibility. And these are all processing parameters that one needs to tease out or test against empirically with different reagents or different pieces of equipment. Some other things to consider is the capacity to scale up the manufacturing process. Some reagents or some automation is not suited for scale up or scale out, and so that is something to consider. In addition, some key points that one should really look into is to optimize your process early, evaluate your raw material components against what your critical processing parameters are, and as well as working with reagent providers early enough to ensure scalability and quality of your materials so that it can be used within a cell and gene therapy manufacturing process. Now, this is a really exciting industry and field, but poses some manufacturing challenges within cell and gene therapy. It's not intuitive, or there's not a standard translational path going from research manufacturing, it's typically done in academia, to GMP manufacturing. In early manufacturing, some don't have defined culture conditions, some protocols are not optimized for scale, and these need to be considered within translating from research to GMP. In addition, some standard products might not suit needs to translate to GMP, so custom solutions might need to be leveraged as a function of that translation. And now when you get up to scale, reagent availability and system compatibility is something to consider, as well as cost control, and again, transitioning your reagents from RUO to GMP with in mind on what piece of capital equipment you're integrating with. Cell separation or isolation at smaller scale might not correlate to larger scale, and automation compatibility is a key consideration that you need to take in with reagents. So as I mentioned before, solving manufacturing challenges using Bio-Techne workflows solutions, we take a unit operation approach. So this might be familiar to some of you. This is a conventional CAR-T or TCR manufacturing process where each unit operation we leverage a different Bio-Techne product to solve a critical problem. Within cell selection and T-cell activation, we have a unique technology, polymer technology called QuickGel that we put in a particle format called clouds to eliminate the needs of magnetic beads and offer differentiating solutions within those two unit operations, including recombinant proteins and activating antibodies for activation. On gene transfer, we've have ACD assets, including RNA scope to profile CAR expression, custom antibodies for staining, and as well as custom CAR design. For T-cell expansion, we've invested heavily within serum-free media formulations and proprietary immune cell medias. For T-cell formulation, we have in-process analytics, including Luminex, Simpleplex, and Quantikine conjugated antibodies to look at different process development steps. We've invested in cryopreservation media, and as well as even after the patient is dosed, we've done application data with Luminex, Simpleplex, and Quantikines to measure cytokine release. In this webinar, I'm going to be discussing the cell culture process development step. Now, as you might be aware, cell culture is full of variables. Within the slide, you can see we've broken it down into 4 distinct variables, including a vessel, media, activation method, and cytokine, and small molecule supplementation, as well as timing. All of these 4 parameters interplay with one another and can affect how those cells perform downstream, as well as how many cells you might get. So some things that I'm going to discuss is optimization of culture media, as well as the activation method used within a conventional T-cell workflow. So optimizing T-cell activation is a critical step within CAR-T and TCR adopted T-cell therapy. Conventional technologies utilize magnetic particles to activate and expand T cells. However, there's challenges with magnetic particles because you have to remove them downstream. That downstream removal causes extra quality steps to measure the amount of particles that you have in your final product. In addition, it reduces the yield of your cell therapy product downstream, and it's also potentially difficult to incorporate into closed systems. So we've recognized the challenge with magnetic particles, and we recently acquired a company called Quad Technologies that had a differentiating polymer called QuickGel that can address this critical challenge. QuickGel microspheres, also known as clouds, can be used to activate and expand T cells, as well as enable non-magnetic cell separation. How clouds works is that you can add a simple release buffer that can nearly instantaneously disassociate that matrix, so you don't have to worry about a magnetic debeading step. We have the ability to customize those cloud particles, making them as small as 5 microns to as high as 500 microns in diameter, and functionalize them with capture proteins or signaling proteins for the particular application of interest. Within T-cell activation, we took the clouds platform and functionalized them with anti-CD3 and anti-CD28 to the surface to make it effectively a synthetic antigen-presenting cell. You can then in turn co-culture them with T cells or PBMCs which then causes the T cells to activate and expand, further expanding over time, and then downstream, simply adding a release buffer, you can nearly instantaneously disassociate those cloud particles and make them suitable for your downstream application, thus eliminating the need for any magnetic debeating. Cloud's CD3, CD28 compares very favorably to existing technologies. In comparison to Supplier 1 and Supplier 2, you can see that population doublings are very similar, but within the Cloud's instance, you do not have to worry about the magnetic debeating. We tracked over day 2, day 5, day 7, and day 9 in population doublings. You can see very similar expansion. The T cells form very nice T cell rosettes, as you can see by the bright field images on the right. External collaborators have compared against conventional magnetic activation technologies in Clouds. In this experiment, they looked at, on the x-axis, a unactivated stimulus of T cells, denoted as 0 to 1, and then a 6-particle to 1 T cell experiment. They demonstrated over the 3 to 1, which they would normally use from Supplier 1, a preferential expansion of early memory phenotypes and different phenotypes, including CD27 and CD28, and as well as CCR7 and 62L, using the Clouds platform. In addition, they looked at T cell transduction of a CAR construct. Again, very similar experiment. They looked at unactivated 0 to 1 and modulated the particle to cell ratio up to 6 to 1 and showed an increase of CD4 and CD8 and CD3 CAR positive cells in comparison to the unstimulated population using the Clouds platform. So to summarize, the Clouds technology enhances cell therapy workflows by increasing the yields, by eliminating the potential cell loss that would occur normally with magnetic particle activation. In addition, it limits the hands-on time, so you don't have to worry about doing a manual magnetic debeating step, and could in turn help with cost savings. So you won't have any residual cell loss, and you won't have to do an extra qualification. Now, in addition, this platform is highly customizable, and you have the ability to optimize for diverse processes and cell types. So by modulating different stimulus, you can drive certain immune cells in different directions. Coming soon, we've invested in humanized recombinant activation antibodies that are stable and from a standardized source, and this product will be GMP-grade for use of ancillary material in cell therapy. Now, in addition to just activation, we also looked at how Clouds played in different media formulations. So the media choice really affects the expansion rates and potential phenotypes of those T cells. So we compared media 1, media 2, and media 3 over a 9-day culture study using Clouds 328 expanded from PBMCs and R&D Systems IL-2, and media 3 is a proprietary media that we're developing in-house at Bio-Techne. As you can see, there was a significant outgrowth of T cells on the left graph, and we looked at CD8 T cell outgrowth as well in comparison to media 1 and media 2. We also looked at the combination of different medias and Clouds as well as different activation reagents. Choosing the right media formulation is integral to optimizing scale, yield, and phenotype, and we modulated different medias with Clouds and different activation reagents with different medias. And as you can see, depending on what media and Clouds you'd use or media and different activation stimulus, you get very different outgrowth of profiles of T cells. Lastly, we looked at GMP cytokine combinations in the presence of different medias. So we compared, again, media 3, an in-house proprietary T cell media that we're developing, and media 1, which is a conventional supplier media, also using Clouds 328 in varying combinations of GMP cytokines from R&D Systems. So in this experiment, we looked at IL-2 and IL-7 and IL-15 over a culture from day 0 to day 12. As you can see, there's significant differences between the media 1 formulation with supplementing of IL-7 and the supplementing of IL-7 in media 3. In addition, within Media 3, the majority of the cytokine cocktails have improved total T cell number and total cell number outgrowth in comparison to Media 1. In summary, workflow process optimization is necessary to achieve scalability and develop a consistent and robust process. Incorporation of novel technologies to improve existing processes can help drive different phenotypes or immune markers that are germane to your process and important to potentially the outcome of your cell therapeutic product. Evaluating raw materials and components in the context of optimizing for yield, phenotype, and efficiency and robustness is an important step within process development. Working with reagents suppliers to ensure scalability, continuity of supply, and quality is a must. Starting with GMP materials as early as possible so you're closer to your final design or closer to your final process. And remembering that quality of raw materials can affect consistency and impact the final product. So ensuring that you're using quality GMP materials will help drive a more robust and consistent process. I'd like to thank you for your attention, and please feel free to reach out to us to learn a little bit more about our cell and gene therapy offerings. Thank you.
Great. Well, thank you very much indeed, Sean, for your presentation, which I think raises some very interesting themes for our panel discussion today. So without further ado, it is actually now time for me to introduce the rest of our panel who will be joining Sean today. First up is Lisa Fox, co-founder of Broken Lance Tech Consulting. Lisa has more than 25 years of experience in development, cGMP manufacturing, and quality control for cell and gene therapy products. She successfully led the development, manufacture, and clinical delivery of numerous ATMPs at Orchard Therapeutics, Sangamo Biosciences, Stem Cells Inc., and Excite Therapies. Joining Lisa today is Angel Herrero Mendez, Head of Pharmaceutical Development at Takeda Madrid, in the Cell Therapy Technology Center for Takeda Pharmaceuticals, where he is involved in the development, validation, and transfer of analytical methods, process development and investigations into technical incidences, deviations, and out of specifications. Next up is Marketa Horopova, Supply Chain Manager at Sutio, where over the last 6 years, she has overseen the procurement activities for the Technical Operations and Research Department for both Czech and Beijing production facilities. And our fifth panelist today is Patrick Ginty, Head of Regulatory Affairs at the Cell and Gene Therapy Catapult. Patrick has 13 years experience in research and development, including 12 years experience in regulatory affairs related to cell and gene therapies and regenerative medicine. So I'm sure you'll agree this is a fantastic lineup of experts with me today, and I'm very much looking forward to the discussion. So without any further delay, I would like to begin by asking the panel to share their thoughts and experiences in terms of the processes that developers and manufacturers adopt in order to select and then qualify raw materials in their bioprocesses. I wonder if, Lisa, you could start us off here.
Sure, thank you. Yes, qualifying new materials, actually any of the materials to be used in an ATMP process is an involved process that involves multiple stakeholders, not just the development group and the manufacturing group who are involved in selecting and then using those materials, but then the quality groups who need to be involved in ensuring that the materials selected are of adequate quality and that adequate raw material testing is in place so that the quality of the material on an ongoing basis can be ensured. So getting the supplier selected is critical. And as a company matures and as a clinical program matures, it's important to have a supplier qualification program that adequately supports those activities. Having good baseline supplier quality and raw material quality established can simplify and facilitate the ability and the need to change materials later should it be needed. And then finally, the last key important stakeholder to involve in this process are your purchasing and warehousing material control groups, because they're ultimately the ones who are going to have to order and ensure continuity of supply of these materials. And so it's very important to get this process right early on and early in the development process.
Yeah, absolutely. I could not agree more. Sean, perhaps you would like to share your perspective here.
Yeah, my perspective on obviously being a supplier, we interact with, a variety of different people within the cell and gene therapy companies, ranging from manufacturers, quality, and process development. One of the big things and challenges are always to get the right people around the room to discuss really what their critical raw reagents are, and what the qualification steps to integrate into some of these processes. So I'm actually curious for the rest of the panel on what their thoughts are on helping corral the right people around the room to have those discussions.
Great, thank you. Angel, would you like to share your thoughts here?
Yeah, so I agree with what has been said, but also I think that it's important to perform audits on the manufacturer, just to evaluate how these suppliers are working, and also to ensure that you have a good supply of the material with the right quality. And finally, you have, as mentioned, to evaluate the materials for its intended use, and when appropriate, testing to ensure the activity or the safety of this material.
Yeah. Okay, thank you. Marquesa, did you have any additional thoughts as well?
Well, from my point of view, it's just to choose the proper material. I mean, if it complies from the GMP point of view is one point, but the other is also if we can buy it in enough amount and at the time when we need it.
Yeah, absolutely. And finally, Patrick, coming to you. Hello, Patrick. Oh, I think you might need to unmute yourself. Okay, well, we'll just move on to the next question while hopefully Patrick finds the unmute. So something that Marquesa, oh, sorry, Patrick, are we there?
I apologize, sorry. So yeah, I was just going to hear what everyone else said, basically. From a regulatory standpoint, it's the same as anything. You're looking at safety, the quality, and the efficacy of the material as appropriate to your process. Obviously, there are certain red flags in terms of things that you might want to consider higher risk, like certain biological raw materials naturally might be more likely to transfer adventitious agents, viruses. TSE risk is obviously slightly higher. And obviously there are things like, people use a lot of terminology and a lot of different terminology in the guidance. So FDA might talk about clinical grade, other regulators might talk about pharmaceutical grade or research grade. And I think the key thing really is to make sure that the material is fit for purpose and worry less about how maybe it's termed, and really look at whether it's actually meeting the requirements you have and whether it's a safe material that's manufactured with a robust manufacturing process and obviously with a robust specification.
Fantastic. Yeah, that's a great point. Thank you. And so let's sort of move on to forecasting, which is what you sort of slightly touched upon there, Marquesa, which it's really such a critical element to effective manufacturer and supplier interaction. I was wondering if the panel could share their thoughts on best practice in forecasting and what specific materials they're going to need, in what quantities they will need them, and when, of course. So, Marquesa, perhaps you could begin on this one for us.
Mm-hmm. I believe that forecasting really is a crucial step in the whole supply and manufacturing process, especially for materials that can directly influence the quality of your product. So, for example, it's raw materials that you functionally test before you approve the new lot for your manufacturer. I think that there are four main factors that can help you with good forecasting or planning. First, it's that you need a real forecast, not KPIs forecast. Second is that you need to be aware of the production capacity of your supplier. I mean, if the supplier can supply you with the amount you need at the time you need. as there might be problem that supplier has, I don't know, limited production capacities, or that, for example, the size of the lot is limited. Then I believe that the third factor is that you need to consider also the shelf life of the material, so it won't expire before you use it. This is important to consider for combination of two ambitious forecasts and short shelf life of the raw material. And probably the last important factor is that you need to know the lot-to-lot variability of the raw material. For me, it means if there's a chance that only 1/3 of the new lots will work in my process, then I will have to be more careful and maybe try to test more new lots at the same time.
Oh, yeah, that's a very good point. Thank you. And Hez, do you have any thoughts around this issue of forecasting?
Yeah, so I will talk from a commercial perspective, and on top of Marketa's comment, I think that it's clear that you need to have a picture of your manufacturing forecast and your needs for your manufacturing in the next month, year. Then I think it's quite important to look at the shelf life of the products because it's critical. Also on your warehouse capacity, just not to avoid problems with your stocks. And finally, I think it's quite important also to look at the quality controls that you have to do. I think that there are some ANSI standards to define the number of units that you have to analyze per shipment that you receive. So the less units you receive, the more impact on your quality controls. For example, if you receive 10 units, you have to analyze 2. You are losing 20% of your units in the analysis. And if you ask for 3,000 units, you have to analyze 50. So this is less than 2% of your units lost on the quality controls. So the higher of units you order, the better for not using a high percentage on the quality controls.
Thank you. Yeah, that's a great point. Lisa, did you have any thoughts here?
Yes, to highlight the special requirements, excuse me, for custom materials, particularly viral vectors or custom media additives. These may also require, when you're doing your forecasting, you really need to take into account the amounts that you might need to do assay analytical method development. You may need to develop the analytical method that's going to be used for the future lot-to-lot variability testing. And then likewise, you may need to develop a surrogate potency assay or functional assay for this material so that lots can be screened prior to release. And then likewise, material will need to be taken into account for stability testing. So sometimes this is overlooked when preparing the material forecast, and then you can come up short. And when you're working with a custom supply of a material that's coming out of a CMO that's already at capacity, getting additional production slots can be quite challenging.
Yeah, that's a very good point. Thank you. Sean, did you have any points to add here?
Yeah. So actually, a few points. So I'll give a little bit of the supplier perspective.
Great.
So from the supplier, when we're dealing with multiple different companies asking for different products, it helps us to understand and work together as more of a partner with cell and gene therapy manufacturers to understand their forecasts. And the reason why is because everyone's competing for very similar products, and without having a robust forecast, it's hard for us to predict when you're going to see an increased demand on certain ancillary materials that we would have to effectively ramp up our production. So prompting, a pretty open conversation around how much one would need with respect to their process is critical for us because it allows us to do our planning for our manufacturing. And obviously, certain products have certain yields, and depending on if it's a lower yield product and if we don't understand a forecast, it puts us in a difficult spot to meet demand when you guys are successful and ramp up. And in addition, it helps with discussing forecasts from the supplier side. It also prompts the quality conversation, and that's imperative to understand, what sort of quality requirements for specific materials that you would like to see, as well as auditing requirements and any sort of regulatory submission requirements that a manufacturer would need.
Great. Thank you. And I guess as well, sort of around this theme, there's often some very unique challenges that the smaller and more specialized cell and gene therapy developers and manufacturers in particular can face when ensuring their supply of critical raw materials. From the sort of supplier perspective, how do you think these risks and challenges can be alleviated, Sean?
Yeah, so some of these challenges can be alleviated in a couple different ways. I mean, going back to my statement before, is that prompting that conversation of having even with small suppliers what they believe that their forecast will be, even if it's trivial amounts within early clinical phases like phase 1 and phase 2, and really understanding that commercial ramp, and so that allows the supplier to understand again their production forecast, as well as making sure that even the smaller players within the cell and gene therapy space get priority based off of what indication they're going after. So I think that's critical. One of the challenges that a lot of small cell and gene therapy developers, as well as just the space in general, is that there are also exclusive reagents that are proprietary that are only from key suppliers, and understanding that as part of master supply agreements that one could carve out certain continuity of supply language to ensure that even a product that is exclusive and unique for a particular process can still ensure continuity of supply. Lastly, the last statement I'll make is that a lot of the small cell and gene therapy manufacturers, because they're tight budgets and they're trying to move from preclinical to clinical, is that they use RUO reagents, and that's perfectly fine for earlier phases. The challenge there is that with respect to RUO reagents, there's not so much in terms of notification if there's a change in that process, and so there's stories of people working with RUO reagents and bringing it through clinical trials, and there was a manufacturing change, and they had to do process development again. So really getting ahead of that from a supplier to customer relationship is imperative.
Yeah, that's a challenge we hear a lot here as well. Angel, I thought maybe you could follow on here.
Yeah, so I think that one of the main challenges where you work with cell therapy products or gene therapy products is that there are most of the reagents that has only an R&D manufacturing process, so it's quite difficult to find GMP-grade reagents for these therapies. And sometimes, for example, if you work with fetal wine serum, there are there are not too many companies with the manufacturers for these serums. So if they are not GMP grade, it's quite difficult, and you only have one supplier. It's quite difficult also to have a backup supplier. So, and it's not only for the serum; it's also for the cell culture media. Most of the media, there are now manufacturers who work with GMP, but most of the media are only available in R&D grade. So I think it's one of the main challenges, and suppliers should be aware that there is an increased amount of people asking for these reagents. Yeah, thank you.
Marketa, would you sort of agree with the perspectives that we've had so far on this issue?
Yeah, I absolutely agree. Plus, there's another one, and it's pricing of the product. Especially when you are a very small company and in the beginning of your clinical trials, you need really small amounts. But especially for the big companies, they are not so much willing to make a reservation of one lot or give you a better price because they really don't trust you. So you really have to persuade them that your product has a bright future and that they want to cooperate with you and have the close relationship with you.
Yeah, great. And Lisa, would you like to share your experiences in this regard?
Sure. A challenge that many small companies, particularly involved at the early stage, don't need to address quite immediately is how you're going to test the novel raw materials. You may be able to accept them early on with the certificate of analysis and the testing that's already provided by the manufacturer. But as your clinical programs advance and your quality systems mature, you need to incorporate verification of the certificate testing and really build robust receiving procedures for the assays. And that's a challenge that small companies buying niche reagents from not always large suppliers can be quite a challenge because you don't get a lot of support from the supplier for these activities.
Oh, interesting. Thank you. And Patrick, we'll come to you here as well.
Hi, yeah, I think it's very challenging when you have very few alternatives out there and you have, small companies basically using what they...
Oh, sorry, we've lost you a little bit there, Patrick.
Me?
Oh, hello? I'm afraid we have lost you a little bit.
Hello?
Okay. Oh, yes, you're back. Okay.
Sorry, I don't know why the sound keeps cutting out. I was just basically echoing what's been said before by Lisa because, very early on, yes, you might be able to use COAs and rely on a very limited specification for the product, but there comes a time when that has to ramp up and you have to start increasing the characterization, particularly as you go through the phases of development, and then looking at alternative suppliers as well. And I think that's where you'll get a more robust assessment from regulatory authorities.
Mm-hmm. Fantastic, thank you. And staying with you, Patrick, we've had a question coming in from our audience, and so it's raw material qualification is performed through risk-based approach, i.e. USP 1043, and then classification into one of the tiers and supplier qualification. For clinical trials phase 1 to 3, we may also do a phase-based approach to have a minimal qualification criteria at each phase. Could you please elaborate your thoughts on this process?
Hi, hopefully you can hear me.
Yes.
Yeah. So, that's exactly really what I was just leading into. So,
yeah.
I think most people would use a phase-based raw materials assessment. So, for a phase 1 clinical trial, you would look at something which is essentially fit for purpose. It may not have a huge amount of characterization. Like I say, the specification might be very simplistic, but normally that's considered to be suitable provided that you don't have any additional risks. So obviously the red flags are things around human and animal-derived materials, and obviously anything that's critical to your process, which is obviously something you define in-house through your quality system. So I think it makes sense to do it in a phase-appropriate way. And then by the time you get to market authorization, it's fair to say that a regulatory authority will dig incredibly deep into the content of your raw materials and, of course, how they're manufactured, because it's important to remember that sometimes materials can be used in the manufacture of another material or in the same facility as another material, which may be a potential contaminant. So there are a number of things to really consider as you go through the phases.
Yeah. Thank you very much. Does any other of our panel wish to comment on that question from our audience member? Okay. In that case, I shall move on to our next question. So, clearly, requiring a unique raw material also adds a level of complexity and risk to any supply chain. What can be done from the manufacturer's point of view to try and mitigate this risk? Marketa, if we could start with you on this, please.
Sure. So, first of all, we should be really sure if the material that we consider to be unique is really unique. Sometimes we are making raw materials unique because we can't decide whether our requirements are like nice to have or must have. So I think that the less unique or custom products or materials we have in manufacture, the smoother the production is. Of course, that the functionality of raw material is always like must-have requirement. So for these raw materials, we need to properly plan the consumption and have a close relationship with our supplier, or start investing into development of our own raw material if it's possible.
Yeah. Excellent. Thank you very much. Lisa, I wondered if you wanted to share your perspective here.
Yes. So while I agree that having unique and custom raw materials adds complexity. Something that small companies sometimes encounter is that working with small suppliers or suppliers of novel raw materials may actually require you to create a custom so that you can ensure that it's made to your specifications. And this is sometimes the case and provides the purchaser with an advantage of then having a unique material that they can then go audit the supplier specifically to see and ensure that the quality of this material meets the requirements for its use.
Excellent. Thank you. I'm just looking at a question from our audience that's come in as well. I mean, quality is something that is mentioned a huge amount in raw materials, and we've touched upon here today already. They're asking, what does the panel define as markers of quality? Maybe, Sean, we could start with you on this one. Hello, Sean?
Sorry, I was on mute.
No.
So there's a lot of markers for quality for raw materials. Depending on what those raw materials are. I mean, for instance, for let's say cytokines to promote immune cell expansion, activity is a huge metric for quality, as well as traceability and whether or not you're claiming a xeno-free or animal-free base material. So it's highly dependent on really what your critical quality attributes for that ancillary material are. And yeah, so and then obviously, when you deal with, maybe not so much cytokines, but if you're looking at a clouds reagent, maybe you're looking for a baseline, functional assay for, activation of T cells measured by flow. So it can vary pretty wildly in terms of what those critical quality for raw materials are.
Yeah. Great. And anyone else from our panel want to share their perspectives on what they would define as markers of quality? I will take that as a resounding no. Okay. So next up, let's now consider the risk assessment and analysis that needs to be carried out on incoming raw materials before they could be released for use in bioprocessing. What are the particular issues here for end users, and how do they change as one moves through development towards commercialization? Perhaps, Angel, you could start on this one.
Yeah, so I think that when you work with raw materials, it's necessary to perform a risk assessment just to evaluate what analysis you have to perform based on the criticity, the occurrence, and the level of detection of the impact of these raw materials in your manufacturing process. And I think it's important also to consider if you have performed an audit on the supplier, what are the observations that you have in the supplier, what is the aspect, what is the manufacturing facilities of this supplier, the traceability of the raw material, what is the documentation which is provided by the supplier, certificates of analysis, certificates of origin, certificates of irradiation in the cases needed. For animal raw materials, you also need a TSA in the case of bovine raw materials. And also sometimes the authorities ask for health certificates of the farms. So you need to have a big control over your manufacturing suppliers, especially if you work with animal origin materials. So then when you perform your risk assessment and you decide the analysis to be performed, you need to verify, according to this analysis, that the specifications provided by the supplier are okay regarding the identity, the activity, the sterility, the presence of viruses. And the analysis that you are going to perform should be validated if you are in the commercial steps. So it's not only to decide the analysis, but also you have to decide analysis that can be validated, and if possible, according to the pharmacopoeias. And finally, after implementation of this analysis, your risk or the level of risk of the impact of these materials in the manufacturing process should be low. So finally, you need to create a sampling strategy and the specification document, the internal specification document for each of these materials.
Wow, it's quite extensive. Thank you. Patrick, maybe you could share your perspective from the regulatory side of things.
Hello, sure, yeah. So obviously, when you're early on in development, you bring through a material, you get things like COAs, and you might send out things like supplier questionnaires. Obviously, for small companies in an early stage trials, you might not go and audit suppliers unless they're really, really critical suppliers. But then as you get through development, it becomes more challenging and you have to provide more evidence. So reliance on the certificate analysis might not be enough. So then you might have to go and audit supplier, for example, as has been pointed out. It depends really on the material you've got and what sort of relationship you've got with your supplier. Mm-hmm. If you've qualified that supplier, you have a very robust material, because one of the things that really kills processes are variable materials. So obviously you've got a lot of that kind of evidence. And if you have that confidence with your supplier, that goes a long way. Obviously, if you have things like raw materials that are authorized as medicinal products, so things like cytokines and human serum albumin, some recombinant proteins, for example, you won't need to provide as much evidence necessarily for certificate of analysis. Likewise, if you have something which has a certificate of suitability to a monograph and a pharmacopoeia. So these things are obviously very advantageous in terms of avoiding a lot of the qualification and risk assessment. But chances are, with most materials in this field, that some level of risk assessment and qualification will be needed, and that gets just more and more robust as you go through.
Fantastic. Thank you very much indeed. And that sort of pushes us quite nicely onto another question from our audience, which is basically asking the panel to comment on the utility and feasibility of the establishment of a raw material supplier certification program for the ATMP space. This would be similar, I suppose, to what other industries, such as the nuclear industry, have in place to help ensure the quality and safety of materials supplied. Perhaps, Sean, if we come to you first of all for the supplier perspective on this.
Yeah, so from the supplier perspective, I mean, we've been following, a lot of at least the regulations within the U.S. as well as some European regulations as it pertains to ancillary materials in the space. I mean, I think it'd be beneficial to have some more standard supply certifications for suppliers. Right now, we do. Depending on really what the field desires, standard ISO 9001, and depending on the material, 1345. That's a requirement. But I think to expand on that, I think would be beneficial for the field for sure.
Great, thank you. And Marketa, I was wondering if you could share your opinion on that.
Well, from my point of view, hard to say. Probably Sean have already summarized it.
Okay, perfect. Thank you. Okay, let's move on to our next question. So I'd like to now ask the panel about considerations around the adoption of new technologies in this sector. What unit operation is the most challenging for process development, and what motivates the incorporation of a new technology? Lisa, perhaps you could kick us off on this one.
Certainly. So adopting new technologies or incorporating new reagents or novel reagents is always easiest during the early development stages prior to having done any GMP manufacturing. We all understand that that's the golden rule. However, we also understand that you're starting early, you need to show benefit or that the program is going to progress before you're going to start to make that investment and advance the quality or move to the more costly reagents. So depending on what the material is and how it's used in the process will increase or decrease the amount of comparability data required to bridge to either an alternate or a new source. And I've started to think about materials used in cell and gene therapy manufacturing processes as being sort of inert and just bystander types of materials versus active ingredients that actually have an activation or an active step. They cause the cells to do something or they do something to the cells as a criteria or as a dividing line between how much work is required to qualify that material and demonstrate that either the change had an impact or didn't have an impact. And understanding that as you progress through phases of clinical development, making these changes will often have an impact on the product quality, and it may be an improvement. And improvements should be viewed as a good thing. And so demonstrating comparability doesn't mean that it's equivalent. You may be making an improvement, and it's how you go about framing that and presenting that and doing the work under a prospective protocol with knowledge and understanding of the change that you're making. So obviously, the need to treat additional patients or being able to time these changes with a logical transition in clinical phase is ideal, but it's not always possible. So these considerations need to be, again, considered by a multifunctional, a cross-functional group. It's not just the people selecting the material and making the change, but the quality and the regulatory groups involved, and then the purchasing and the warehouse folks who are involved in stocking and storing those materials.
Yeah, absolutely. That's a really interesting perspective. Thank you. Patrick, I wondered if you wanted to comment on this.
Yeah, so just to pick up on the comparability issue, which Lisa was talking about, obviously, as you get through development, it does increase, and you know, regulators will look at clinical trial applications or INDs, and first and foremost, they're concerned with safety, so they want to see comparable safety, the data, and then obviously you get into phase 3, your pivotal studies, you're going to be measuring efficacy of the product, therefore the functionality of the product then becomes into focus even more than it has done previously. So it's never too early to characterize your raw materials, particularly if they're critical. So like, you know, we've alluded to cytokines, things like interleukins, which are often used in T-cell processes to activate cells. These kinds of materials, really, you need to understand exactly what their impact on the functionality of the product is, and obviously on the safety profile earlier in development, more importantly, because then when you do have to make a change or you have to have an alternative supplier as you get towards BLA or marks authorization application, you really want to understand what the impact is going to have when you do those comparability studies. Otherwise, you might find yourself with, you know, potentially a product which is just as safe, but potentially doesn't have quite the same efficacy profile as you had previously.
Yeah, that's a great point. Thank you. And so, I mean, Sean, what are the considerations for introducing raw materials into closed automated systems, which we hear so much about the industry moving towards?
Yeah, I mean, this is something that has been fascinating us from the supplier side, because frankly we didn't realize how much of a challenge it is just introducing ancillary materials to culture vessels. And so what we've been looking at in terms of challenges in the current workflow and just simply adding additional growth factors to a process is that those are typically done in dilutions, are done in isolators and then carried out by an operator. And obviously that could pose some challenges and risks to your therapeutic product if you have contamination or operator error, or you have to do extra qualification. And so the thought here is that, you know, what if you can introduce these ancillary materials in a closed weldable fashion, so you don't have to, that's already preloaded with whatever dose of growth factor you'd like into a closed system, just by a weldable container closure. So I'm actually curious on what the other panel thinks on that challenge in introducing ancillary materials into closed systems.
Yeah, absolutely. So Angel, would you like to comment on this question?
Well, I have not so many experience in closed systems, but I think that most of the comments that we have done regarding the materials for the open system should be applied in the same way for these closed systems. What I could mention is that if you are moving, sometimes when you move from the open system to the closed system, you also want to change the culture media. And I think that here the importance of good tools for evaluating the comparability of your product is critical.
Yeah, thank you. Okay, I think we have time for another question from an audience member. So this one is around the fact that with limited cohorts for a number of these new cell therapies, we are seeing accelerated movement through the clinical phases and in some instances movement from phase 2B to commercialization. Did the panel envisage that with these fast tracks, we'll see a larger number of marketing authorizations being rejected based purely on the robustness of raw material data provided to the regulatory bodies? I wonder if perhaps, Patrick, you could start us off here.
It's an interesting question. I think the likelihood of raw material stopping a product being licensed, I'd say, is probably quite low, because you'd expect something that was really critical to be picked up, even in a relatively fast development program. You'd expect it to be picked up fairly early on in the process. I think where you'd more likely to run into trouble is if you have introduced something very novel and then made changes. Obviously, one of the biggest things you can fall foul of potentially is cell culture media, because particularly when you're manufacturing things like pluripotents, the cell culture media tends to be very complex, and it may be that you don't fully understand the early implications of all those components within the cell culture media in early development, and only during late phase development you will understand them. So while the risk is there, and I say for certain critical raw materials like cell culture medium, it's probably higher, I would imagine that the biggest worry would be having a backup supplier. Really, it'd be more of a commercialization risk than a regulatory risk. So it's more having that ability to replace the material or to supply when the supply you rely on can't provide it for you.
That's an interesting perspective. Thank you. I wasn't sure if any of our other panel members had a take on this question. Perhaps Sean?
Yeah, I mean, I think Patrick hit the nail on the head on this, and it's a lot of these issues would be addressed when they file their first IND, and so I think the risk is fairly low because diligence has been carried out on the supplier. But to Patrick's point, your limiting step could be on the commercial side of making sure you have A, continuity of supply, and B, a redundant supplier just in case something happens.
Yeah, absolutely. Okay, well, sadly, we're getting close to finishing up here, but I think we have time for one more quick question. So what can be done to accelerate the qualification of a novel raw material or technology with one's bioprocess? Perhaps, Lisa, if you'd like to just kick us off with this one before we go around the panel.
Certainly. Well, I would like to quickly echo some of the comments and perspectives provided by the other panelists earlier on. It's never too early to start qualifying your suppliers and gathering as much information on every material used. Having all of that documentation from the beginning can greatly facilitate the ability to make changes later, because while at the end of the day you still have to or do the risk assessment and determine the requirements to make the change and then do that work. There have been, and many of us have endured recall situations and urgent situations where the entire field has had to make substitutions or adapt to supply situations. And so careful evaluation of the material in use, certainly doing some head-to-head testing and ensuring product quality and doing a risk assessment of all the potential areas for product impact and then monitoring for the possibility of those situations occurring become important things that can be done to just have control of your current situation so that when the time arises to make a change, you can move as quickly as possible.
Yeah, absolutely. That's a great point. Angel, if you come to you for this as well?
Yeah. From my point of view, to accelerate, I think that it's quite important to have good standards in your process. I mean, if you have a cell product, if you have a standard for your product, reference standards, you can compare your new your novel raw material with the current ones that you are using. And I said something which is simple, in fact, but it's not so easy to have reference standards for cell therapy materials. So I think this is one of the challenges of the next years, have good reference materials in order to ensure comparability of raw materials or whatever change you are going to do.
Yeah, absolutely. And Patrick, coming to you here.
Yeah, so in terms of novelty, obviously there's always a little bit of a nervousness around novelty and the word novelty when it comes to anything to do in the manufacturing process. I think really it depends what the material is and how critical it is to your process. But sometimes it's impossible. Sometimes there just won't be a material that you can use which is available. And from my perspective, I would feel more comfortable going to a regulator with a novel material which is more fit for purpose, but doesn't perhaps have the great breadth of data behind it, but perhaps using something which is available, but doesn't actually suit the purpose of the needs, and further down the line might actually do you more harm. So sometimes maybe it does pay to be a little bit brave if you think there's something you can do which is going to make your manufacturing process and therefore your product safer or have a better quality or a better efficacy.
Thank you. Marketa, I wonder if you had some final thoughts here.
Yeah, I believe that it was summarized by Lisa, Angel, and Patrick. From my point of view, the power is in information that you have about the raw materials. And I absolutely agree that you have to start collecting them as soon as possible.
Yeah, great. And then finally, Sean, for you to wrap up.
Yeah, no, I just wanted to echo Lisa's comment. It's never too early to talk to suppliers. You know, wherever either if you're in preclinical or early stage process development, just understanding what the critical needs are as we go on is really great for us, as well as us to communicate with people who are manufacturing. I think fostering that open relationship between the supplier and the cell and gene therapy manufacturer is a must.
Absolutely. Wonderful. Well, sadly, we have run out of time today. It just seems to go so quickly on these webinars. But thank you so much indeed to our panel for a great discussion today. And thank you to our audience members as well for your questions. Sorry if we weren't able to get to your question today. We'll be passing this on to the panelists and hopefully getting back to you in the next few days, especially some of the questions that you had for Sean regarding some of his presentation as well. So we will pass that on to Sean directly as well for follow-up. So yeah, that leaves me to say thank you very much today to Sean, Angel, Lisa, Marketa, and Patrick for joining me today, and thank you to our audience. The on-demand will be available tomorrow, so do look out for an email from me. And yeah, we look forward to you joining us next time. Thank you very much indeed.